Disability worldwide, and these are data from 2016. Worldwide, 268 million people suffered from an episode of major depressive disorder, and that's about 1 in 5 people over a lifetime. So this is very, very common. But it's not just a question of it being common, it's also a severe disorder. And there are 800,000 people who kill themselves every year, and that works out about 1 every 30 to 40 seconds. So in the US alone, it's about 30 to 40,000 people will kill themselves this year. And half of those are because of depression. So this is common, and it's killing people. And we're not particularly good at treating it so far. So currently we're spending about $10 billion on 245 million prescriptions for antidepressants. And we're doing that even though I would expect, as a psychiatrist, only about half of my patients to show any sign of improvement after about 3 months. And this situation is made even worse by the fact that over the last few decades there have been no novel or more effective antidepressants that have been put forward. So we have a common disabling condition which we treat badly, and as scientists, I'm sure you'd agree the first place to start to change this situation is to try and find out what causes depression. And here we have one clue, and one really only solid clue, which is that it has a genetic basis. And I just want very briefly to review why we would take a genetic approach to identify the causes of depression. And I think we can just summarize this very simply, and I'll do so in the following couple of slides. So when we look for causes, we look for things that are associated with the disorder, stressful life events, immune system disorder, changes in neurotransmitters. There's a whole list of these that people have assembled over the last 50 or 60 years. But in so doing, all we have is correlation, and we don't really know what causes what. Whereas a DNA sequence variant associated with the disease must precede the disease onset because it's fixed at conception, and therefore the DNA sequence variants actually give us causal information. And that's fundamentally why we tackle a disease like depression at the level of the DNA, because we can get at the cause. So for those of you who haven't thought about this before, just very briefly, remember that you have 2 chromosomes, one from your father, one from your mother, and occasionally at certain positions along those chromosomes, there are sequence variants indicated here by that red letter. And occasionally those have functional consequences. So for example, I'm showing you here one in a gene, and let's assume that it alters the structure or function of the gene in some way. The 2 variants are called alleles, and because there are 2 of them within a population, we can expect to create 3 genotypes. You can have the homozygote A, the homozygote T, or the heterozygote in between. And in a genetic association, we simply look to see whether there's an enrichment for one or more of those classes within the disease that we're looking at. So for example, we might ask, are the people who have depression associated with those with a homozygote, and those who are unaffected with a T homozygote? And that's essentially all that we have to do in a genetic association study. And when we do that, we apply a statistical test and come up with a p-value. So if we do that, then obviously we'd like to know just how to interpret that result. And just so you understand how these things work, there are a number of interpretations. The first is simply we got it wrong. That we have a p-value which doesn't reflect a true association. And that's not a trivial issue. It's dominated the field for a long time. We think we know how to deal with that now by setting appropriate thresholds. The second possibility is the one we're looking for, namely that there is a causal relationship between the variant and the disease. And that's like the example I showed you, where we have a variant lying in a gene, and we can say that alters the structure or function of the gene, and that causes the disease. But there are 2 other possibilities we need to consider. The first is that the alleles at the marker locus are correlated with those at the disease, but they don't directly cause the disease. And to show you how that might be the case, just look at the figure below. And here I'm showing you some sequence information again, 2 chromosomes, and there's a T and G in red. Let's call that the causative locus. And a bit further along, very close by, there's another sequence variant, an A and a T. And it may be that that's the one we've genotyped, and we've got information about that, but we can't distinguish whether it's the A and the T or the T and the G. They're just too close. They're highly correlated. And then there's a fourth reason, a fourth interpretation, another reason why we might not be finding the causal locus, which is there's something called population substructure, where the marker allele frequencies in subpopulations correlate with the disease status. And just to show you how that might be the case, I'll give you one slide. So here I'm showing you the cases and controls, and there's a risk allele in red and a protective allele in black. And these have been sampled from 2 populations. And if you look under the box of cases, you'll see that population 1 is overrepresented. And because population 1 happens to have more of the red allele, if you do a comparison between the cases and controls, you'd come to the erroneous conclusion that the red allele is the one that's causing the disease. In fact, that's not the case. It's simply because we've sampled more of population 1. In order to deal with this problem, we go to people like Jennifer Lisgarten and Eliezer Eskin, who've thought about this and come up with some computational methods that will deal with it. And again, we think this is a solved problem. So we have those 4 outcomes from the genetic association study, and if we carry out an association, we might find something like the following. So the way to interpret a figure like this is to look at the vertical axis, which represents in a negative logarithm to the base 10 of the association statistic. And each of those dots you're seeing is the result of a single variant that's been tested. So there's maybe 1,000 along that locus. And you can see in the middle there's a peak, and there's a couple of markers which are at about the value of 10 to the minus 6, and then there's ones having less significance below that. And we would conclude, if we apply some threshold, that that's the place where we're seeing a signal which is associated with the condition. And the only difference between this and what we do in practice is instead of just looking at one place in the genome, we look at markers throughout the genome. We do it genome-wide, and we get a figure that looks something like that. And this is for a study of type 2 diabetes. And you can see those peaks. The peaks give this plot the name of a Manhattan plot, which indicate the position on the genome where we think there's a locus affecting the disease. And notice there's a dotted line, a horizontal dotted line, running at about the level of 8, which represents an accepted level for the significance threshold. So any of those places that exceed those threshold we regard as being evidence for a locus associated with the disorder. So now you know how to interpret a genome-wide association study and all the complexities that go with that. I'm going to go and show you another slide where you have to tell me, or at least for yourself, what the results are for depression. Pretty bad, eh? So this was the case until about 5 years ago, and when people saw results like that, they really asked the question, why has it been so difficult to find the genetic basis of depression? And the first reason that a lot of people considered was very simple, that it's a psychiatric disease. And as everyone knows, psychiatric diseases are very hard to study. And there are a number of reasons why we know that's not the case, and I'll give you perhaps the most obvious one, which is a result from a study of schizophrenia, classic psychiatric condition. So there, you can hardly see the significance threshold. It's so low down on the plot because so many of those peaks are exceeding it. It's quite clear you can find lots of highly associated variants for a psychiatric disease. So it can't just be because depression is not a psychiatric disease. A second possibility is maybe it's not very heritable. So schizophrenia is a relatively heritable condition, between 60 to 70%. What about for depression? Here's the evidence that depression is actually a heritable condition. These are results from twin studies. There are 6 very high-quality twin studies summarized here. The horizontal line is the variance in the estimate, and the little vertical tick is the point estimate for the heritability. In the bottom, you can see a summary, a meta-analysis, and that indicates that the heritability is about 37%. And that's pretty consistent with diseases like type 2 diabetes. And I showed you a result earlier on for type 2 diabetes where we found lots of genetic effects. So it's heritable, sufficient for us to be able to expect to find loci. So the next reason which has attracted most attention is because each genetic effect is small. And if each genetic effect is small, that would mean we'd have to study a very large sample in order to see an effect. And maybe it's simply a question of power. Maybe just we didn't have a large enough sample. So a few years ago, this study was published. This is from a consumer genetics company, 23andMe. And you can see they've highlighted in red where they've got significant loci. And I've put in nice capital letters underneath the sample size of 75,600 cases and almost 250,000 controls. So really dealing with the issue of sample size in an effective fashion. And then last year, this study was published from the Psychiatric Genomics Consortium, putting together a massive 135,000 cases and 344,000 controls, and now we're up to 44 loci. And if you go online and look in the bioarchive, you can find this study, which has now gone up to almost 1 million cases with 102 independent loci and replicated in 1.5 million, a massive study. So you might think from this that this problem is therefore solved. It's just a question of sample size. However, there's this single study which I was the director of. We can see we have 2 loci there, which exceed our threshold. Not particularly impressive given what we've said before. But this study consisted of a tiny number compared to what you've seen before, a mere 5,000. So that's inconsistent with the results I showed you before. So why might this be? And we've considered some possibilities. Maybe it's just wrong. Maybe there's some population structure. Maybe there's some other features going on, something we haven't considered up to now. So the first thing to do is just to do a simple comparison between what was found in one study and what was found in the second. So I'm going to take the loci from the Converge study, that's the smaller study, and I'm just going to compare the significance of those with the those found in the 23andMe study. So I'm going to look in the 2 studies and see. So at the top, what we are seeing there is the significance of the 23andMe, and then we're looking in the sample size in the Chinese study, in the Converge study, and you can see that it trundles along the bottom, and there's really no sign of an effect there. So what we're really asking is, if I found something in the large 23andMe study, does it replicate in this other study? And the short answer to that question is it doesn't. There's just no indication of any signal whatsoever. So you might argue, well, maybe that was just power, but we can, for various reasons, rule that out, which I won't go into. And let's therefore turn to look at something else, which is what exactly was being measured. So how do we diagnose depression? And if we go to the 23andMe study, it's very simple. So they were asking some questions, which I've listed out here. Have you ever been diagnosed by a doctor with any of the following psychiatric conditions? Yes, no, or I don't know for depression, and so on. So the diagnosis of depression was based on whether you told the company that your doctor had diagnosed you with depression. That really doesn't work if you're a psychiatrist. Like if you come to me and say, my doctor tells me that I've got depression, I'm not going to accept that from you because there are many reasons why that might have happened, and I don't really know whether you've met the criteria. So the study that we ran, we had a 1.5-hour interview with you, each of the subjects, to determine whether you met what is known as the Diagnostic and Statistical Manual, the DSM criteria for depression. We also focused on a subgroup only of women with recurrent depression recruited through hospitals in order to really isolate a group where we could not only be certain that they had depression, but it was a severe recurrent form of depression. So this focuses on a very critical debate that goes on in the community between what I'll call a minimal phenotyping, did you ever have depression, yes or no, as against a deep phenotyping, come and talk to me for an hour and a half and I'll decide whether you have depression approach. And there are advantages and disadvantages to each. So obviously the minimal phenotyping is quick, it's cheap, it's scalable. I can run this on 1.5 million people by asking that question, whereas the deep phenotyping is slow, expensive, it's not scalable. But the fundamental difference between them is, what are they measuring? We know for definite that the deep phenotyping measures depression. That's how it's diagnosed, that's our gold standard. But we don't really know what the minimal phenotyping is diagnosing. So we've looked and we found a population where we can compare the two, and we'd really like to know, are we measuring the same thing or not? And the answer to that question in short is no, it's not measuring the same thing. And I'll just give you a couple of bits of information as to why I say that. So the first is that we can take a series of risk factors for depression. The risk factors we know include sex, include environmental life factors, age. There's another set of risks involving whether you take drugs, you smoke and so on. And we can look at each of those risk factors and ask, how do they perform in these two diagnostic categories? If what we're measuring is the same, then of course we'd expect the pattern of risk factors to look the same, but they don't. So I'll show you first the results from looking at a traumatic life event. Traumatic life events increase the risk of depression. And here what you're seeing at the top, two different DSM, that's the deep phenotyping based diagnosis of depression, and I'm showing you the odds ratio and the p-value for that association, and underneath are 4 different ways in which we have of measuring depression through something like a symptom-based approach. I'm just simply asking if you have the symptoms of depression. A self-report, did you ever have depression? Help seeking, did you go to your doctor? Did he or she say that you have depression? And what you can see is that there's a highly increased odds ratio for the DSM-based diagnosis compared to the help-seeking, the minimal phenotyping approach. So the risk factors differ. We can produce similar patterns if we look across different risk factors. But of course, what we're really interested in is the genetic underpinnings, because we want to get at the cause. So just because the risk factors differ doesn't necessarily mean that the genetics is different. So now what we're going to do is going to look at the genetic correlation between these different forms. So here on the vertical scale, I'm plotting out the genetic correlation between a deep phenotyped diagnosed measure of depression and the others. So on the left-hand side, there are 2 different DSM-based, that's the deep phenotype approaches, and not surprisingly, they basically are the same. So I'm getting a correlation of 1. And the other 3 are these minimal phenotyping approaches, and there the correlation drops down to about 0.8. So there's some genetic sharing, but it's certainly not total. So all of that comes around to saying that really a critical element that we have to deal with a disorder like depression is how it's diagnosed. What is the phenotype? And the problem that we have here is that it's still a diagnosis made by a trained doctor, and it takes a long time. How are we going to get over that problem? How can we make it more scalable, make it more appropriate to run on a study which has sufficient size really to detect large numbers of genetic loci and understand what is the genetic architecture? So this has been a major focus of the Depression Grand Challenge. The Depression Grand Challenge is a campus-wide initiative which has been running at this university for the last 3 years. And we've set as our goal understanding the causes of depression as a first step towards curing it. And because it's Los Angeles and we're in California, we're not just going to cure it in LA, we're going to cure it in the entire world. And we've set ourselves this target. We're going to do half of it by 2050, and we'll get rid of the entire disorder by 2100. And that gives me enough time to resign and presumably retire and maybe not even be alive, so I won't ever be accused of failing in this. But this is our aim, and we're pushing towards it. So as I've said, a core element of what we do is trying to work out how to diagnose and assess depression in a systematic, efficient way, while making sure we are diagnosing depression and not some minimal phenotype measure. So the problem we face is that the tools that we use really haven't changed for 100 years. We have some questionnaires, we have diagnoses based on this DSM criteria, but as it says there, they're subjective, to some extent they're biased, they're certainly time intensive, and they're static. We have one data point. So the solution that we've been pursuing is to try and use digital methods, to try and use electronic records, to try and use what we call a behavioural health tracking app. We want to put something on everyone's phone which is going to give a measure of their mood. And to do that, we want to measure lots of different things about you. We want to measure your voice, we want to measure your activity level, and we really want to try and drill down to those measures that we know from lots of other pieces of work assess mood, which is things like sleep, activity, concentration. And what we suspect is that a lot of that information is encoded in an objective fashion in things like what you do with your phone. And if we had enough information, then we could presumably take all of those data and allow us to make predictions about your social interactions, sleep, physical activity, and mood, and allow us to turn that into a diagnostic category. So we've got as far as a prototype. This is obviously a collaborative effort with many different groups, and a group in Finland running a software, an open source software called Aware, have come up with a method that does this. So if you were to open up your phone with our app on it, it would, for example, be giving you these little surveys, which where you enter some data and it'll work out roughly what it thinks your mood is, and then it's going to be quizzing your phone, taking off various bits of data to try and associate that with the more objective measures that we've got. And we've been picking up things, and I'll just show you one very brief piece of data to give you a rough indication of what we might be doing. And this is a measure of your activity. So we're just going to interrogate where you are based on GPS coordinates. And here's a picture to show you the sort of data that we might hope to get, which is that a simple measure of the percent spent at home, and you can see in this particular person that it correlates very nicely with their depression severity score. Of course, I've cherry-picked. There are lots which don't show that, but it's at least an indication that there is some signal in the data, and if we work hard on this, that we'd expect to turn this into a much more robust and objective measure, and I should really emphasize an objective measure. So the expectation is that using these devices, the connected measure, we can accelerate our research, and most importantly, that we'd really be expecting us to be able to understand the biological causes by integrating it with the genetic data and ending up with a real insight into what causes depression. But it's not only just getting at the cause of depression per se. There's a lot of data to suggest that this is not one disease. I won't go into all of this, but we have to work on the assumption that it's the common outcome of many different pathways. So one of the things that we'd hope we'd be able to do is by taking the range of data we get off the phone and from the questionnaires, is that we can identify subtypes of disease. And in so doing, we'll be in a much better position to develop a much more precise measures of treatment. There is a rather specific problem that anyone working in this area trying to find the causes of depression faces, and that is the following. We have now a way of assessing your mood from your phone, and what we expect to do is to give this out to lots of people, collect the information from them, and make diagnoses and try and understand the cause. What we will find inevitably when we do that is half of the people who we diagnose as depression will not be receiving treatment. And there is no way we can mount a large study, and by large I mean we're looking at maybe 100,000 people. There's no way we can mount a study of that size, make a diagnosis of depression, and particularly if amongst them there are people who are telling us that they are thinking of killing themselves, there is no way we can do this without offering some treatment. I told you at the beginning of the talk that the treatments we have are not very efficient, but that doesn't remove from us the obligation of at least trying as best we can. And you've also heard that we think depression is syndromic, many different forms. We would hope that as we progress, we'd improve in targeting the treatments we have to improve the ability to treat those who are part of our study. So not only do we have to develop the methodology to collect data about the disorder, at the same time, we have to have these connected devices to actually aid treatment delivery. So they have to do two things. They have to make the diagnosis, and we have to be able to go out there and start treating people. Now, this has some interesting implications. One of them is the following. Typically, if you come and see me, I will sit down with you, we'll have a chat, I might make a diagnosis and change your treatment, and I'll ask you to come back in a week's time and then go through the same process, and then there's a third visit. And after that, for example, some crisis will happen and you'll have to be admitted to hospital. Now, I, as you can see from that graph, would have no idea as to whether that actually was going to happen or not. And that's because I'm seeing you at these time points. I don't know what's going on between those. If I now give you an app measuring your mood, I'd hope to change that graph into something that looks like this, where I'm taking measures in real time, which will give me a much more precise indication of how your mood is changing, so that I should be getting alerts from the machine saying, hey, this person really needs your attention. Get them into hospital fast, start some treatment. So the first thing we should be able to do by tracking your mood interactively is giving whatever treatment's available, if you're already in the treatment system, much more early. And we should be able to prevent the sort of problem that you can see here, which is of a crisis occurring without background help. But we can go one step further. Not only can we provide help from existing sources, we also want to be able to treat directly from the phone. And we do that by integrating the phone with a system of interactive cognitive behavioral therapy. So typically a lot of psychological therapy is given now by sitting down with a psychologist who will train you in certain ways of thinking which we know will improve your mood. And what we can now do is automate that system and provide it over the phone. So the way this would work is as follows. You get an app on your phone, it includes a screening system. No evidence of depression, that's fine. If there's evidence of depression, maybe an indication that you're suicidal, then we'll start a tiered system. So those who we regard as mild, we now give an online cognitive behavioral therapy, and I'll show you an example in the next slide. A moderate will get that, plus some support over the phone with a trained counsellor. It doesn't have to be a psychiatrist, but someone with some training in this area. And maybe that might be a week or more frequent, depending on the severity. And those who are really severely ill, certainly those who are threatening to kill themselves, will have to come into the standard psychiatric service. For those who are getting the online cognitive therapy, we are currently using a system developed in Australia called This Way Up, which is a series of cartoons. It's been validated. It performs just as well as seeing a psychologist, with the big advantage that you can do it anytime, any place. And on the phone, it'll be sending you reminders, tracking your progress, and if there's a failure, also alerting your therapist to try and improve. So just to summarize how this whole process works, I can put this in the following slide, which is we have an integrated model. So we've integrated the research and the treatment into one single seamless system, where we're screening you with an app on the phone, we're analysing those data, we're coming up with a diagnosis, treating appropriately, and then tracking and monitoring you in real time. And that system has now got through its an initial stage, and we've been using it on the student population in UCLA. So all undergraduates have been having an email asking them to come to take part in this study, and if they agree, we put the app on their phone, and we've been providing them the treatment. And in my last slide, I just want to show you very briefly what the results are. So this has been running now for 18 months, and since the launch, we've, as you see, screened over almost now 5,000 students. 740 have been identified and have signed up for the research. And of those, there have been 143 suicide risk alerts and 187 severe depression alerts. And the feedback we've got has been consistently positive. What's really proved attractive to this population is the fact that they can do it, as I've said, when they want, where they want. If they want treatment at 2 o'clock in the morning, they get it. They don't have to see anybody. It's great. Really has been working very, very successfully, and we've had a lot of positive feedback. And I'll finish there. There's a huge list of people behind this. The Depression Grand Challenge is, as I said, a campus-wide initiative, but not just this university. We have colleagues in Northwestern, in Chicago, in Finland, and our colleagues in Australia who've worked with us on the internet CBT system. So I'd like to acknowledge all of their help and take any questions. Thank you very much.